A couple of announcements as we move forward. So as you may be starting to think, we have a midterm coming up, okay? And I know that we've received a few questions in office hours and so forth. So I just thought I'd take a minute or two to briefly describe it, as well as answer any questions that you might have. The spirit of which is I don't want you to get nervous or stressed or anything, so forth, okay? So it's next Friday, right, in class. So it'll be, 50 minutes in class. And first of all, it's open pretty much everything, okay? Not open neighbors or maybe open internet, but you can feel free to bring your computers or however you manage your notes, anything, books, so forth, okay? The spirit of the test is not, it's a short test, right? So the spirit of it is not to have it be so detailed and so forth that you'd need to refer to all those other materials. But I sort of like open book. Hopefully you do too. Just that way you're not worried about, trying to memorize this or cramming it into a little sheet that. The format's typically, in the 5 to 6 question range, right? So that we cover a variety of things. The coverage will be up to and including handout 8, meaning homeworks number 1 and number 2. And last time we finished handout 8 and so forth, okay? The homework, which is due next Wednesday, I believe, in class, right? We'll have the solutions for you so that there'll be a couple of days to, check to see if you had any questions and so forth. Okay, are there any questions? Am I forgetting anything? Yep.
I'm curious if there was anything of a really technical issue today. Was the email about what we were supposed to read for today actually sent out at 8:56 or?
There was an announcement that came out, but I'll talk about that in class, and it's totally optional reading. It has nothing to do with the midterm that we're talking about.
Okay, thanks. Okay, I just want to make sure that I wasn't supposed to.
Yeah, we'll get there, yeah. Okay, so midterm? Okay. So we're switching gears here a little bit. We've really covered, the biophysics of neurons and the statistics of neurons up through handout 8. Handout 9 that we'll talk about today and next time really is about measurement, right? Sort of a return to, let's look at some pictures and think together about how you evoke action potentials in the nervous system, how you measure them, and sort of, there's not, a great deal of intellectual content, but it's more context setting so that we have a good chance to talk in class about how these things are done and so forth, okay? And then starting handout 10 and 11 and so forth is really we're moving into decoding as we've foreshadowed, okay? All right, so the reading assignment that goes along with lecture 9 is a PDF that's posted on the website by Rutan et al. And there's no one definitive paper that covers measurement start to finish. This is now a few years old, but some of the newer things really go into more super detail, and it's I think more important just to sort of have a general overview of how these things are done. Okay, this also marks the first paper that we're reading, and by paper I mean paper in the so-called primary literature, right? So when research is done, papers are published. That's how colleagues are kept informed of what's going on. Eventually, if that becomes believed enough through time, that could enter textbooks. That's what we've been traditionally reading. And as we move forward, we'll be reading a few of these primary literature articles, right? And that's the spirit of that is that they're carefully selected. There will be certainly parts of them that will be probably too challenging, but most of it will be readily approachable. And again, the spirit there is just to start introducing you to that world as well. Many of you are already familiar with that, but it's sort of fun. I mean, that is the end-all, be-all. These are papers that are coming out every few months from different research groups. Okay? All right, so what will we talk about in handout 9? Well, selective electrical interfaces with the nervous system require approaches at the size scale of the neurons, okay? And we've talked that neurons are, between 1 and maybe 50 microns in size. You can think of that spherical soma area. Let's just call it 10 microns to keep a rough number in mind. Okay? So that's pretty small. It's not super tiny, right? What's a modern gate length on a transistor?
45 nanometers, 60 nanometers, something like that.
We're talking nanometers, right? Yeah, tens of nanometers, right? Which is 3 orders of magnitude, right? Minus 9 down from micrometers, okay? So it's not as though we're accustomed to building things far smaller than neurons, okay? The interesting point is that we can't possibly wire up a transistor to as many other transistors as a neuron is wired up to other neurons. And we can't pack them in 3 dimensions like the brain is and so forth. So there's many tricks biology has to overcome the fact that it's building things that are a little bit slow, a little bit sloppy in terms of information processing, and a little bit large. But it works out. Okay? So neurotechnology, okay, which is sort of a buzzword in the past decade or so, is really at the intersection of neuroscience and microtechnology. So think about the various fabrication techniques used in microelectronics, silicon, patterning that silicon, etching that silicon to make very small structures, putting metal on silicon to make transistors or oxides and so forth, okay? Doping the material with different conductive elements, p-type, n-type, and so forth. And the neuroelectronic interface is really where the membrane meets, and we'll call it a metal electrode from now. It can mean a whole variety of other things, but basically if you take a small electrode and you put it into and outside of a neuron or even into the neuron, as we've seen many times before, right, with things like patch clamps and so forth, that interface is where a lot of the action is, okay? Now that interface, the exchange chemistry and so forth around there really is complicated, okay? I don't like using the word complicated because it sounds like, oh, we can't approach it. But it really does require a considerable amount of time, so we're not going to go into that in any detail. Okay? But just please know that there's something very interesting going on there. Okay, so electrode design for either stimulating or recording, and we'll talk about those two in turn here, requires a good understanding of the membrane, the nature, the natural and evoked action potential generation process, volume conduction, in other words, how do currents go through 3-dimensional volumes of solution, right? Largely saline, sort of a saltish water. That's what our central nervous system fluid really is. Okay, and the electrode behavior. And what we'll really be looking at are a whole variety of so-called penetrating multi-electrodes that are produced either in linear arrays, right? Just sort of lines of these electrodes, 2-dimensional arrays of them, or even 3-dimensional. And mainly they're made out of glass and silicon and metal technology. And the reason for that is that those are the materials we like. Those are the materials that we're largely familiar with. It doesn't mean they're the right ones, that they're the ones that are going to win in the long run, because just because they're nice materials to run through a fab and Intel and every other company knows how to do these things, doesn't mean that it's really all that groovy when it comes to putting them next to squishy neural tissue, right? These things are rather stiff. Those things are rather pliable. I think we've talked about the idea of taking a pin and sticking it into Jell-O, where Jell-O mimics the brain. It's not a great idea, but it's getting us a long way. Okay. Other types of electrodes include cone ingrowth electrodes that we'll talk about, and this is actually where I'll sort of take a brief aside that meshes with the announcement that came out. There was an article that appeared in the literature. Actually, I shouldn't call it literature. I should be specific. It appeared in Wired magazine, which I distinguish harshly from real literature. Okay, and I want to spend a few minutes talking about the extremely important issue that could also be an entire class and is, but neuroethics. Okay, how should one think about, well, we can build a bunch of stuff. We have the technology, but what should we do with it? How do we responsibly do it? I'll try to describe roughly what that is, and then we can contrast that perhaps with what this article reports. Okay, it's timely. It came out on Tuesday, I think. Okay. Okay, we talked about this, including growing neurons on top of electrode arrays. Now, that's not something that's going to give you a natural nervous system, but it'll allow you to study it. Okay. So overall, it's important to be familiar also with the signal flow in neural data acquisition systems for neuroprosthetics. So in other words, once we have these electrodes in place to stimulate the nervous system or record, let's talk also towards the end of handout 9 a little bit about how we take those signals and process them. And you'll have an opportunity to do more of this in homeworks. Okay? So does that give you a sort of a whirlwind overview of what we're going to dive into? Okay, so let's think about stimulating the nervous system. Okay? Not recording from neurons, but causing action potentials to fire. Okay? And on the first and second lectures of the class, we talked about one example of this, and that was when an electrode is placed into a deep brain structure called the globus pallidus, an electric current is trickled in to stop Parkinsonian tremor. Do you remember this example? So you're putting electric current in. It must be causing action potentials. It could also be suppressing them. Another example is artificial vision systems. Take a camera that can see this room quite wonderfully, take the video output, translate it into different patterns of electrical stimulation you should apply across, for example, a two-dimensional array, and have that array embedded in the retina. So even if your retina no longer has photosensitivity, like hopefully most of ours in this room do, to see directly, you could electrically stimulate the nervous system to serve as a proxy for that visual information you take in. There too is an example where we need to cause action potentials. Okay? And really, a theme in all of biology, and we see it rearing its head here too, is always selectivity. Okay? It's not good enough just to sort of hit a general area and activate a million different neurons, because that would sort of write in some garbled message. You really want to try as hard as you can to stimulate individual neurons or small groups of neurons that might be doing similar things. Okay? So that's just another theme to keep in mind. Now, let's consider the case where the local extracellular application of a depolarizing, okay, in other words, cathodic, monophasic, okay, that means just a single pulse, short, about 100 microseconds, current pulse in the microamp range may excite an action potential, right? So this is one of those super dense sentences that we have to read like 8 times to piece it all together, okay? But basically what we're talking about is putting an electrode outside of a neuron, okay, applying a current, so that positive current's going into the region just outside of the cell body, which will tend to depolarize that membrane, right? And if we're depolarizing it, we increase the propensity for it to fire an action potential, right? And that can be done with very short pulses. They needn't be long, 100 microseconds, and they need not be large, microamps, right? The type of thing that you would barely feel if you don't do this, right? But if you were to take a microamp current generator and sort of apply it to your tongue, you might feel a little sensation, but it's not much, right? Again, don't try that at home. All right, I can't help but resist it. Can't help it. Has everybody licked a 9-volt battery in their childhood at some point? Yeah, I mean, how, right? That's probably highly correlated with the fact that you're double E's, but that's a. Anybody licked an outlet? Don't try that at home either. Okay, you didn't hear that. We'll erase that from the video. Okay, now when you do, okay, so let's look at this plot here. Okay, now there's a lot going on. First of all these dash curves, ignore those. Put those out of your mind. And let's just look at this solid curve labeled A. And this is the transmembrane voltage change, or the change in the membrane potential, right? Okay, V out minus V in is the membrane potential. 0 is there's no change. And this is time. So let's come in and put an electrode in and stimulate it with 80%, 0.8 so-called IT, the threshold current. So there's some minimum amount of current that you would need to apply that you could find out empirically to cause this neuron to depolarize enough to fire an action potential. And let's come in just below that. Let's come in at 80% of that level. So we won't fire an action potential, right? And that's what you can see here. When you turn on that current pulse, it does change the membrane potential. And then you turn off that current pulse, presumably here, and it relaxes back to having no change, right? So you've transiently perturbed the voltage on the outside of the cell. It did cause, of course, a membrane potential change, but then it returned itself without firing an action potential. Now, if we go up in current a little bit more, just barely to threshold, that's what you see in B. You hit it just hard enough now that it ticked it just here. Maybe there's some uncertainty. It sort of like wobbled around a little bit. Boom. Then it fires this action potential as shown here. Okay? And what would one expect if you came in at, for example, 20% above threshold, as shown in curve C? Well, here you kick it. It comes up so much above the threshold for firing an action potential, right, that the positive feedback loops kick in. Remember these sodium and potassium channels, right? Okay. Boom. Here's an action potential, and it fires. Okay. So you can just simply do this. It's really as simple as, if you had one of these giant squid axons, you just put an electrode there, and you just sit there with your current meter. You do this experiment, and there you go. And then you could, of course, answer what threshold current is. Maybe it's 120 microamps, or maybe it's 48 microamps. That depends on the exact geometry of where that electrode is. Okay? Yeah?
Why the sharp peak and trough for the one right at that?
Right here? I don't know for sure, but it must have to do with the fact that you're driving the cell's external potential up to this point. And then it may take some time, okay, for the positive feedback loop to kick in and really continue it on up here. And in the meantime, it's just doing some little delicate balance between trying to restore it, but oh no, now the channel's kicked in, now it kicked it up. So there's usually some little kink right around there. I'm sure the dynamics can be fully explained. I just don't know them off the top of my head, and it would be a sort of a detailed calculation. Okay, so big picture, right? You can put an electrode outside the cell. You don't have to see the neuron and impale it. You can just put it right on the outside by passing current, right, that flows through the solution. And you can imagine that solution having little resistive elements. Current going through those resistive elements changes the potential. You change the memory potential, you fire an action potential. There you go. That's the fundamental basis for how you would write information into the nervous system, right? Video camera stimulates these electrodes, stimulates neurons in the retina, creates artificial little colored or oriented so-called phosphines, little glimmers of light on the retina. And there you go. Or Parkinsonian tremor can be stopped by activating these neurons that way. Now, what are the dashed curves? The dashed curves are simply saying that if you go up to, a cell, or let's consider an axon, okay? And let's say that the electrode is closest to, for example, one node of ROM-VA, okay? And you stimulate it, you'll create an action potential there. Now, if I give you a symmetric axon and I depolarize the membrane right in the middle, which way does the action potential go? Left or right or both directions? Both, right? So you're kicking it right in the middle. There's absolute symmetry. There's nothing causing it to go left or right, so presumably it goes both ways. Okay? Now, in practice, maybe it goes one way or the other, but in practice, it does go both ways as well. Okay? That's really the right answer. The only reason we think of action potentials only going one direction down an axon is that they're usually generated over here. And once they start propagating in one direction, they can't reverse on themselves because the refractory period in their wake, left right behind the wave passing through, will cause it to not be able to depolarize going backwards. So there's no chance of an action potential spontaneously flipping on itself, okay, without a weird setup that might show up in a midterm. There are ways. Okay, so an action potential, it's meant as a Friday joke, but nobody, okay. It's okay, I'll cry myself to sleep again tonight. Oh, there's the sympathy I was looking for. Finally, okay, thank you. All right, so it travels in both directions, right? There was a little clue to my question, I suppose. And this example of the membrane change in response to external stimulation is what we've really been talking about. Okay, so how to think about this in slightly more detail. We've already foreshadowed a lot of this. This isn't hard. Okay, a negative going extracellular current pulse close to a node, for example, of RON-VA here, may trigger an action potential artificially in the central or peripheral neurons. Okay, typo. Modeling this is usually done in 2 stages. First is nerve fiber excitation, and the second is volume conduction. Okay, now we're not going to do anything with this. This is just sort of cultural exposure, right? But you can imagine having a simulation package, okay, that really takes into account where the electrode is. You could come up with a model for that electrode, which clearly it's metal, so it's going to conduct, but has some finite resistance, so there's probably a resistor equivalent in there. And then the tip to the solution is usually modeled as a capacitor. Okay, and that's why you have to have these little current pulses. You have to have some time-varying change, otherwise that capacitor is just an open and nothing happens. And then you can bring it up next to something that should look familiar, which is some distributed model of an action potential, right? Here's the myelin, here's a node, the axon's running down right in the middle, these are the nodes of Ranvier, and we have some equivalent model, right? We have our channels, we have the membrane capacitance, we have some axial resistance. Remember these types of models from several lectures ago? Okay. So you can imagine putting in a current pulse, boom, in time through here, how that current would spread all around here, ultimately returning to some ground terminal that you'd put in the system somewhere. And when that current flows, the potential along those current lines is changing. It's actually quite linear. It's amazing. This extracellular fluid is conductive, it is slightly resistive, and it's extremely linear. If you double the current, you'll get double the voltage drop. One wouldn't necessarily know that, but it turns out to be quite linear. Okay? And then you can model in 3 dimensions how it changes the voltage everywhere here, and you can understand how it could depolarize it. Now, once you have that depolarizing event here, okay, and you understand this first part, this nerve fiber excitation shown in green, the volume conduction part of it is, again, as we studied before. If we're thinking about propagating down an axon, remember we were very sensitive to the diameter of it, the capacitance set by the walls where myelin wraps itself around it to reduce the capacitance, and how the voltage propagates down this, which is the same as understanding how it propagates down one of these myelinated sections, you can do with other types of simulation packages. Okay? So just like for transistors in circuits, you have a whole suite of simulation packages that you probably haven't directly worked with yet, right? Probably everybody's worked with SPICE or familiar with SPICE, a circuit level model, but understanding how exactly electrons are moving around in super tiny spaces is the domain of other types of simulation packages, okay? Same thing. It's just another physical system, electromag holds, away you go. Okay, selectivity. At low current, the electrode can stimulate one fiber meaning like an axon, okay? If its position is closer to that fiber than to other fibers. So it's really very basic. Put the electrode closer to this axon than this axon, and you'll preferentially stimulate this one. And that can be important because if you stimulated both, maybe you're conveying a mixed message to the next part of the brain, and it'll be hard to interpret. Okay? Now, as the increase of current will expand the stimulation volume, and therefore you'll include more and more fibers. So you also have to keep that current low. Remember we were talking about a roughly 100 microamps for an electrode that's fairly close to a neuron or an axon. If you turn up that current too much that's going through the electrode, then the current paths through that solution outside will spread farther and farther, meaning it'll have voltage drop farther and farther, and you're more likely to activate this over here. So not only does the electrode need to be close, the current needs to be kept low in order to have selectivity. Okay? Does that make sense? I don't think there's anything terribly complicated. It's just saying how do you target. Okay? So ultimately, the selectivity is reached if each fiber had its own electrode. And that would be ideal. But it's very, very difficult. No system yet achieves that. Okay? Such a situation would require both enough electrodes and a blueprint of these neuron fibers in 3-dimensional space, right, that you could position those electrodes perfectly or optimally. No blueprint like that is yet available, okay? And microfabrication has its technological limits. Does that mean you can't take out neurofibers and section them and look at them under microscopes and say, oh, well, there were 18 different nerve bundles and here there's exact locations? Of course you could. But you need to do that in the animal when it's intact and then also deliver the electrodes to each one of those. That's very difficult. You couldn't just make an electrode array in the fab and come over here and stick it in. Okay? You'd have to actually have that spacing all worked out. Or you could say, well, what I'm going to do is I'm going to make a super high density, like a hairbrush, where each little tine on the hairbrush is an electrode. I'm just going to stick it into my neural tissue. And so some electrodes will be very close to one, and other electrodes will be close to the other. Yeah, you could do that, and that would be quite good. And then the electrodes that are in between, you just choose not to stimulate. You could do it that way. What's the problem with taking something like that, though, and practically trying to do it? So imagine the brain is Imagine the brain is Jello. Okay, and imagine not just putting in one needle, but imagine having a whole bunch of needles, like a bed of nails, right? You know the bed of nails thing. Take a board, put in such high density nails, you put it upright, and then you can walk barefoot on it. And it's like it's a little creepy. Why does that work? Why doesn't your foot just get impaled?
Yeah.
Go ahead.
Distribution of pressure.
Distribution of pressure, right? So if you have enough contact points, then there's not enough force or pressure on any one electrode to actually penetrate through. Flipping that around, if you took a chunk of brain, imagine like Jello, you took a bed of nails that's so dense, you couldn't push it in without ripping that tissue apart, without ripping that Jello apart and just mushifying it. It's a technical term. Mushifying, right? You'll destroy it. It's too high density. Said differently, slightly more scientifically, you're displacing too high a fraction of the volume of that tissue. Okay? So if you take a chunk of Jello and put in one needle, no problem, because the needle is pushing the Jello out of the way, but there's a whole bunch of Jello on all sides to absorb that compression, right? But if you start putting a needle in every 10 microns, okay, there's no place for it to all to go. It'll start ripping. Okay, so in practice, it's not good enough, and this foreshadows the coming few slides, okay? In practice, it's not good enough just to make something that you think should work. You have to think all the way through in these medical systems through actual surgical implantation, reliability, how the implantation problem, okay? How would something survive for a long time, okay? Hey, what material do surgeons put in when they replace a hip? Or when they need to, if you break your jaw, for example, too badly, they can put in a little strap of material and put some screws in, right? What material is that made out of?
Platinum.
Platinum?
Or titanium.
Titanium? It's titanium. Platinum could work for various reasons, but you'd probably go broke. Yeah. Titanium ain't cheap. Think tiny little screw, think $100, you know, for titanium. Why titanium? Titanium has just been found to be just pliable enough, inert enough, doesn't rust, you know, has all these different material properties, and that's just to put a screw in bone, right? Now we're talking about having to understand materials and geometries to put into pliable tissue. So there's a whole set of design rules there that we're not going into, obviously. I just want to help you appreciate that there is a really interesting challenge there. And that's really the domain of a lot of what goes on in bioengineering in the department here, throughout the medical school. You know, lots of ideas. It's sometimes called tech push, right? Here's the trap we never want to fall into. We as engineers say, oh, I know your problem. I'll just make something small. I know how to do that, right? And you build it, and you love it, and you think it's going to work, and you walk across the street to the medical school, and you get laughed at. And you should get laughed at, right? Because you didn't appreciate the problem, the fundamental problem, okay? The problem of how is that going to be delivered? How is that going to get maintained and so forth? So that's really where interdisciplinary work and collaborations come into being. You always have to know the problem, right? And at the same time, the responsibility goes the opposite direction, right? Clinicians and surgeons deeply need to know the material systems as well. Okay, and that's the type of thing that goes on a lot at Stanford. Okay, let me not dwell on that more. Hopefully you appreciate that problem. There's a big open space there. It's not as simple as just saying, I'll make 10 more transistors here and make a circuit and somebody will buy it. Okay? All right, so we must test the degree to which stimulation can be selective and the extent to which each electrode controls as few fibers as possible at as low current as possible is very good. And that's important before the potential fields start to overlap with those of other electrodes. That's that current spreading and therefore the potential spreading. Okay? A greater overlap always means less selectivity. Question?
As far as having a very high density of electrons, at some point, do the electronics themselves lead to too much heating for the brain?
Yes.
Does this occur before or after the point at which your displacement is too great?
It depends on the packing density of the electronics, all the other operating characteristics of the electronics, and so forth. Yeah, it's a great question. So far what I've been talking about is just the electrodes, where you could imagine then tiny little wires taking those electrical measurements outside of the brain. But let's say that on the actual, and we'll see pictures of this in a second, on the actual electrode you actually put electronics on the backside. Electronics are powered, they heat, right? We're accustomed to heating issues on our cell phones and our laptops. We don't like heat because it burns battery power unnecessarily. Heat's a real problem in the nervous system because you're slow cooking tissue, right? So I'll give you a rule of thumb. I won't ask any questions because it'll take too long. But it turns out that about 1 degree Celsius increase is believed to be about the maximum increase in heat that nervous system tissue or the brain can take on a permanent basis. Okay, so if you put an electronic payload onto the surface of the brain, it can't heat up more than 1 degree C. Now there's some good news because you have a lot of vasculature that can transport some of that heat away. It's like an active cooling system, right? Our body is all about extracting heat and not causing overheating, right? Right? Ever go for a run and just sit there and double over and go, you know, you're halfway panting, right? Gotta cool down. It's all about thermal management too. But that's a pretty harsh restriction on the electronics that you would build, okay? Especially when you're doing it at density. Okay, so what do some of these measurement techniques look like? Okay, now this is sort of a busy plot, but it's done for a very particular reason. And that is that on the vertical scale here, you're plotting the spatial resolution. In other words, how small a feature in the nervous system can you measure? Okay, and on the horizontal is that's log millimeters. This is log seconds. This is time. How quickly can you measure it? Okay, so let's just explain this by looking at some examples. Okay, and by the way, dual labeled because log millimeters is sort of a hard thing to think about. Over here, here's the size scale of a synapse, a neuron, and the whole brain of a human or of a monkey. Okay, and same thing here. Here's milliseconds. Here's a day, a month, a year. Okay, now patch clamp, something we know about. You go ahead and grab onto a cell, right? Well, you're measuring an individual cell. That's pretty much a small feature, the best resolution you'll ever need. Okay, unless you're measuring channels and so forth. Okay, so that's why the spatial resolution here is really small. You're measuring down at the level of the neuron or synapse, and the time resolution is phenomenal as well. You're dealing with sub-millisecond right in here, but you can also hold on to that recording for maybe a few minutes. And that's why this is shaded out. Does that make sense, why it occupies that region? Okay. Now up here, let's just consider for a second EEG. And this is where you put surface electrodes on the brain. Okay. And this is done all the time in neurological clinics for detecting seizures or other sleep disorders, this type of thing. Well, there, you're measuring not down this small. You can't. You're on the outside of the skin, on the outside of the skull. The electrical signals that you measure are really the aggregation of thousands or millions of neurons. So you're washing out a lot of information, okay? But you're able to measure really across the whole human brain. You can put on a cap, and you can get these measurements across. And that's hugely important, too. There's no right or wrong on this plot, right? It's just showing the complementarism of all these different methods. Okay, magnetoencephalography as well. Non-invasive imaging methods, MRI, fMRI, occupy this space, right? Which is giving you better spatial, better temporal resolution. Here's a single electrode, right? When we drop an electrode in just outside of a neuron cell body, right? You're not able to record from the synapse of the dendrite, but you're recording from a given neuron. That's why it's up here at this level of spatial resolution. And temporally, you can record, for many, many, many hours. This is done routinely. You can put that electrode in, just hold onto that neuron for 6 hours, 8 hours, if you wanted. Now, what this big shaded region has really been the focus of much of modern research for the last couple of decades. And this is called chronic, meaning permanently implanted, multi-site, meaning you can put electrode arrays a few different places, electrode arrays. And here's a picture of one of the workhorses in the field. Okay, this is a little bit outdated. The company's now called BlackRock. But basically, this is a silicon micro-machined array. This is 4 millimeters by 4 millimeters. Here's a 2 millimeter scale bar. And what you see is 100 of these electrode arrays, of these electrodes. There's a 10 by 10 grid. And they're each, although they're ominous looking at this resolution, they're only 1 millimeter long. Okay, I mean like 1 millimeter. And what you do is that you can put them on the surface of the brain and then push them in ever so slightly. And the tips of each one of these electrodes comes to rest just outside of a neuron, just like this. And you can record all of a sudden from roughly 100 neurons at the same time. And if you didn't just put one of these arrays in here, let's say you put another one in over here, okay, you could all of a sudden have 200 neurons. Or maybe put 2 more on the other side, 400 neurons. So you're able to measure from individual neurons, i.e. retain this edge down here, very small size scale, but you're also able to measure across the brain, and because they're permanently implanted, you can measure individual action potentials, but you can do so for years. Okay? That's what we, for example, use in our lab. Many labs do that. What is going into human participants as part of FDA clinical trials that you saw in the first couple days of class where you're thinking about moving something, this is the workhorse of that. Okay? There are so many limitations of this. The quality doesn't hold up over time, ... But circa 2016, I want to leave you with a clear impression that this is typically what's going on to access individual neurons, but to do so over years. Okay? So here's a couple more pictures. We've already looked at this one. You can imagine making the same electrode array, but with a slant. Okay? And this is all just done by various different etching techniques and masking techniques, just like goes on all the time over in the Paul Allen building, right there. Okay, and the the rooms with the yellow lights, because that's where lithography patterning goes on, right? So you literally can build these right there. We don't, but you can. They have been done so in the past. Professor Greg Kovacs, up to maybe 15 years ago, made millions, or not millions, but thousands of electrode arrays, probably millions of electrodes, okay? Also different shapes. You could imagine a single shank here where there's different electrode tips along the length. And these are so-called one-dimensional arrays. You can see different pictures of it. And you can imagine how you could even insert that into a peripheral nerve, like we were talking, where maybe each one of these dashed lines is a different nerve bundle. And by putting it in here, it looks sort of horrific, like you're slicing it. And, well, to a certain extent you are. But maybe you're damaging only one fiber, but you're able to record from 2 or 3. Okay? But then you have to think about mechanical stability of that setup. All right. Here's another picture where you can take a bunch of these electrode arrays that are along lines, and then you can start assembling them, putting them under caps. Here's an example out of the University of Michigan. And you can keep building these up so they look more and more impressive. I guess this is the key one. So all of a sudden you can imagine how in one dimension, two dimensions, right? Because you can record from the tips just like that other array I was showing you. But now you can also record up the shaft as well. So this is a so-called three-dimensional recording technique. You're measuring from neurons in a whole three-dimensional volume, like in a little sugar cube. Okay? But delivering them surgically has turned out to be a real challenge. So, you know the old saying, if you build it, they will come. Well, if you make it, it won't necessarily be used, right? So it's that reminder that we were talking about before. Okay, so the final slide, and in our final 10 minutes, this is where I wanted to, oh, sorry, question.
Going for the previous slide, what was the issue that they can't be used?
Oh, I don't mean to say that they cannot be used at all. The issue that, sorry, thank you for clarifying. The issue is that you have many, many delicate, potentially brittle structures. And we were talking before about if you get this density so high, you have this bed of nails effect, and you'll not just nicely insert this into the brain like you'd insert this into a piece of Jell-O, but it might start slashing it. Or once it's in there, even if you do get it in there, because when you just, for example, look at my head here, that's about a 5G head turn. Okay, staggering, right? And what your brain is doing in there is it's just, right? It's compressing and moving. Okay. Now, if you do that much faster, right? If you all of a sudden, well, I'm trying to get you to lick outlets and smack your head into walls today, apparently. But if you just crashed your head into the wall here, you'd probably get roughly a 10G acceleration, right? And you don't do that because what do you get immediately? Concussion, right? Okay, that's your brain inside the skull just going compressing and then slapping back and rattling, right? Potentially causing microbleeders. Now imagine doing that with that in your brain, where you have this stiff, rigid thing, okay? So even if you're putting it in, for example, a laboratory rat, okay, or a monkey, let alone a human, you have to really be thinking about that mechanical safety as well. So these reasons of delivering it into the tissue, displacing too much tissue and causing tearing, safety due to mechanical, this is a stiff material inside of a flexible material that needs to be able to mechanically adjust itself, right? It's a mechanical impedance mismatch is what I'm describing, if that makes sense. Okay? Yeah?
So when you're trying to pack more electrons into one single square area, doesn't that also introduce greater issues of ISI as well?
Sure. If you're recording from more than just one neuron on an electrode, then you'd have to be very careful about separating which neuron you're recording from, so-called spike sorting problem. And at the tail end of this lecture that we'll pick up next time, we'll actually be talking about that a little bit. Not about the detailed algorithms for doing it, but I'll point out the categories of algorithms that do it and highlight exactly what you're describing. Okay, so in our remaining 5 minutes here, let me show you one other technology which is very cool, okay? And it couples into the article that we sent a link out to. Okay, now what you'd really want is something more futuristic, and here it is. I think this is really a very interesting design. It's been around for almost 20 years now. It's called the cone electrode, okay? And it was invented by Phil Kennedy at Emory University, okay? It's been used in monkeys and in humans, okay? And here's an example of when it was used on a human clinical trial participant who was so-called locked in with Lou Gehrig's disease, unable to communicate. So what they did was they neurosurgically implanted a tiny glass electrode, okay? Just imagine a super fine, like, pipette. And inside of that, they put a little bit of sciatic nerve harvested from that same patient. And why put some sciatic nerve? Because it exudes so-called neural growth factor, okay? It's basically a chemical signal that says to nearby neurons, so here's neurons of the brain, that it's good to grow into that cone, okay? Right, so neurons can grow, they can move, right? They're constantly pruning, meaning throwing away little synapses here, or growing other synapses. The only thing that doesn't tend to happen is you don't tend to grow brand new neurons, okay? but neurons can change their shape and grow. So this is really brilliant because what you're doing is you're basically saying, okay, here's a code. I'm going to send out a signal and have things grow into me and form a really stable connection. It's very clever. Okay? Now, it's hard to do that at scale necessarily, but if you put in one or two of these electrode arrays, or one or two of these electrodes, you make these long-term connections to these neurons. Wires come out through the skull, and then you have a power induction coil, right? So it's like your toothbrush at home. You're powering it through induction. And then you get a transmitter out here. Now, a lot of this is done in many other systems. This is really the unique part of what Professor Kennedy's been doing. And here's a radiograph, a lateral X-ray, okay, showing the receiving coil. Here are the two electrodes. Here are the electronics just below the scalp, and so forth. Okay. Now this is very cool. Now, what in the last 3 minutes here do I want to point out? This also is the first time that we're really looking systematically as we're going forward at a system sitting inside of a person. Okay. And the ethics of doing this is really very important to always have front and center and to keep in mind. And the general way it's supposed to work is the following. Okay? And the way I can guarantee to you that it works at Stanford and all other major universities is the following. Okay? A new technology is conceived and it's worked out in laboratory animals. Okay? Laboratory animals are, of course, very important for testing of pharmaceuticals or other devices. Any medicine or surgery you've ever had or undergone was tested that way. It has to be. It has to be tested that way to make sure it's safe. When there's enough data, then you go to the Food and Drug Administration, called the FDA. They review all that data extraordinarily carefully, right? It makes commercial electronics, FCC, and so forth not look so impressive. It's really rigorous because you have human safety at stake. If it looks encouraging enough, then there are multiple stages. If it's a pharmaceutical, there's a phase 1, phase 2, phase 3 trial, right? So anybody that enjoys the stock market or biotech or anything like this is keenly aware of what happens. Lots of so-called clinical trials, very carefully regulated, extremely high oversight to always assure the participant, the volunteer, of safety. Often the volunteer doesn't know if they're getting the treatment or not. So-called randomized design, because you have to know whether it's working or not. And placebo effect is a real effect, right? If I just tell you I'm giving you something, but I give you a sugar pill, you will feel better, guaranteed. It's pretty cool. If you can trick yourself into that, it's not a bad way to go, right? It's like talking yourself out of bed when you don't feel well. Okay, that's the flow, carefully regulated. Here's what should not happen. You shouldn't come up with an idea, think that would be pretty cool, fly to somewhere in the world where you can convince somebody, by paying them perhaps, to stick it in your brain. And then definitely what should not happen is it should not be written up in the popular press such that it confuses people and potentially hurts fields, because it's just fringe. It totally sidesteps and gets around this very careful covenant between the government, between people, between physicians, between patients, between researchers, between societal good. Okay? So the article I sent out is in Wired by Phil Kennedy, this individual. Okay? And it's very easy as a scientist, right? I mean, unfortunately, there's many movies, right? The mad, evil scientist who gets discouraged by the slow rate of progress and decides to just take a shortcut. Well, read the article and judge for yourself, okay? So I don't want to say too much more, but I want to describe this for a couple reasons to make sure that at least in broad picture I describe how the careful bioethics, neuroethics in this field are handled, how from animals to people, the regulatory environment is really something you can believe in, okay? You can believe in it, you can feel proud in it because it's protecting people, okay? And when something hits the market, we know there can still be side effects and drugs can have to be pulled off the market or devices, but the best possible thinking is done, right? And you can be doubly proud. The head of the FDA right now is a Stanford graduate, in fact, okay? The article that you'll read will challenge some of your thinking. It will, at some times appear quite reasonable. Well, of course you should be able to do this. So I challenge you and encourage you to read the article just for fun. Right? There's never going to be a question on this, right? This is just for fun. This is beyond the classroom and trying to get you to be, critical thinkers, as we like to promote at Stanford, right? And just think about it. Ask me questions. Ask any of us questions if you have, and we'll go forward. All right? Have a great Friday and weekend.